Good morning. Honorable Speaker, Dr. Sushmita Dev Ma'am, Founder Head of Zoology Department, the Assam Royal Global University, Professor Dr. Nikhil Kumar Churungu, Dean, Royal School of Life Sciences, members of faculty and compeers, I extend you a warm and gracious welcome to this august gathering. I am Disha Agarwal from MSc Zoology, 2nd SEM. It's an honor to be your host for today's event. We have gathered here to hear intently from an eminent educationalist, Dr. Sushmita Dev Ma'am, Founder Head of Department, Royal Global University, and currently working in the Department of Biomedical Engineering at Cleveland Clinic. Now, I would like to invite Dr. Dharmeshwar Barhoy, Assistant Professor, Department of Zoology, to deliver the welcome address.
Respected Dean Sir, Professor Nikhil Kumar Churungu, respected Sushmita Dev Madam, and all the other faculty members and dear colleagues and my dear students. I, Dr. Dharmeshwar Barhoy, on behalf of Department of Zoology, with great pleasure and enthusiasm, I welcome all of you to this interactive session. This interactive session actually organized by the Department of Zoology for the PhD scholars and the other students to motivate them towards research. This interactive session represents an invaluable opportunities for all of us to broaden our knowledge, to perspectives, and to encourage all of us for this to interact with this interactive session. And I believe that with this discussion with Dr. Sushmita Dev Madam, after this interactive session, it will emerge with new ideas and the perspectives and strategies that will benefit for the department and the other and as a whole broader academic that is community. As we are there towards this interactive session, I encourage each and everyone to actively participate in this interactive session, and also to try to clear your doubts and give your valuable insights. I extend my gratitude towards Dr. Sushmita Dev Madam for sharing her valuable time for this interactive session. And once again, I welcome all of you. Hope that this interactive session will gives us with new inspiration and growth for all of us. Thank you.
Thank you, sir. We are fortunate to have Dr. Nikhil Kumar Churungu, sir, as a beacon of guidance and inspiration. I request Professor to kindly do the honor of delivering the keynote address for this session.
It's not only a keynote, it's just a little bit of notes. Good morning, everyone. What do you find in this display here, which is most, you can say, prominent, or over which your eye goes? Can anyone tell me from the students? Anyone? None? The ever smiling Sushmita. Isn't it? That is the prominent face that you have over here. It's a pleasure to be here. I know her because when I joined here, she was already here as an associate professor in the Department of Zoology, and very enthusiastic personality, which would always tend to work towards the growth of the department. Here, sitting here, she amongst us, within us, has two aspects. One for this, from my point of view, one from this, for the students. That if you join the Royal Global University, what's your aim? Do you have an aim of doing your masters or leaving the university after BSc and or leaving the university after MSc and then settling somewhere for a job because the background of Royal Global University may not give you a good value to your degree? Or is it that you say, okay, MSc हो गया, अब शादी कर लेते हैं, बात खत्म? Or is it that you want to pursue your dreams and you have set your dreams, which could be anything, but higher than what you are doing now? Sushmita is an example of that dream which all of you should have. She worked here for how many years? I think 5 or 6 years. Here?
Here,
5 years.
4+.
4+ years. In the 5th year, she got an opportunity. She must have been trying for that opportunity. She got an opportunity and she took advantage of that opportunity to pursue her dreams abroad in Ohio, Cleveland, Cleveland. And for the teachers, for the faculty, the message is that don't think that you have become an assistant professor or associate professor and that is enough. You have to work towards achieving good goals. So the goals are set for both. And it's not that sitting in RGU you cannot pursue your goals. You have examples, you can pursue your goals provided you have set your targets. And most of the time what I see among the students especially, their targets are not defined. They don't know what they want to do after they do MSc. But that leads us nowhere. I welcome Sushmita here. I call you by first name. I hope you won't mind.
No, sir.
Right? And it's a pleasure to have you here after 1.5 years. 1.5 years. When she left, a few days before leaving, I still remember she called the Vice Chancellor and we in the Zoology department. Do you know there is something in Zoology department laboratory by her action, because of her? Do you know? Students of Zoology department? Louder? Aquarium. And why is it that the fish are dying in the aquarium? Even before she was to leave, when normally people would think, "Arey, ab to mujhe chhodna hai, mujhe kya jana hai." She gave a gift to the Department of Zoology that was 2 aquarium, I think, 2 aquarium, and that was a remembrance. That shows her linkage, her association, her fondness for the department. She came to India back after 1.5 years and she came here delivering a lecture. And what I would expect, that when she goes back, she takes with her 1 or 2 students from this place. Or she guides 1 or 2 students to go and pursue their dreams there and achieve something much better than this place. Or if that's not possible, because that has to be two-way traffic, students also have to be good enough, hard-working enough. At least she discusses with some of the faculty members here to have joint collaborative programs, and there is already a call from National Science Foundation, DBT. If you people are interested, you can have a program under that. But it takes time and it takes effort. So effort has to be there. She has the background, she has the training, she is here to help you all. That's one part of the story. The other part is her talk. That will be on hepatocarcinoma. How many of you know what is hepatocarcinoma? Good. So you have a background of this, and she will be talking, I assume she will be talking about the latest information on the therapies that are used to treat hepatocarcinoma, because that's a very dicey disease. It's very difficult to get rid of a hepatic cancer. So she will be able to establish her, I am sure she will be able to establish her footmark here as well as in her place. She is working there as a postdoc fellow. And once you are in US, in an academia, if you are good, I think you are good forever. We were telling her when she would go, "Arey, ab kya yahan aana hai?" She would, "Yeah, I don't think." But she made it a point, she came here, we welcome you, and we are thankful to you for your interest in the department and talking and interacting with the students. Thank you, Sushmita, and we welcome again.
Thank you, sir. We are always grateful for your guidance. Moving forward, it is my immense pleasure to introduce our speaker, Dr. Sushmita Dev ma'am, who is a distinguished academic with a vast array of experiences spanning decades. Dr. Dev earned her PhD from Guwahati University in 2016 and began her career as a guest lecturer at Cotton University. Ma'am conducted a notable clinical project on depression and prolactin at Guwahati Medical College. Rising to the position of Associate Professor and founding head of the Zoology Department at Royal Global University, she led a significant 2-year study on the prevalence of liver disease amongst different ethnic groups of Assam, addressing critical healthcare challenges. Currently, she is leading 2 pioneering projects on hepatocellular carcinoma in the Department of Biomedical Engineering at Cleveland Clinic, solidifying her reputation as a leader in the field. Without further ado, I request ma'am to kindly begin her talk.
Good morning, one and all. Present here, respected Dean Sir, our senior professors, faculty members, and my dear students. A few are very known face to me. I'm getting emotional to see all those faces and many new faces. And I'm very happy to see the department growing under the leadership of our respected Dean Sir and all the faculty members. So when I came and I went to the laboratory, like this department is like a brainchild to me. So immediately I first went to the lab and I'm very happy to see that it's growing. And thank you, Churungu sir, for your guidance. And I'm because like Dharmesh is here, he's a very good researcher. He has got good number and all about the new faculties I don't know much about, but today I'll get time to interact with the faculty members. So once again, I'm emotional and I'm happy to be here. So like, let me begin with my presentation. And I would like to add, like when Disha was telling that she's from the Department of Biomedical Engineering. So my lab is a collaborative lab between biomedical engineering and interventional radiology, which is the clinical aspects. So I have 2 aspects of my research, biomedical where we do the biological researches and the clinical aspect that is in the Department of Interventional Radiology. So I would like to give an overview about what is hepatocellular carcinoma. So since my audience is PG students and UG students, I would like to start from the basic of liver structures. So with this I begin. So we all know that liver, it has 2 lobes, right lobe and left lobe. So this is the basic structure of the liver, which is the largest lobe. So right lobe is always larger than the left lobe. Okay. Now these are some of the cells of the liver, and if you get into when we go into the physiology of the liver cells, we see that there are certain important cells which are hepatocytes, which we call as the liver cells. Then there are Kupffer cells. Then there are hepatic arteries, bile ducts. So these are some of the important features of the liver. Now coming into the main blood supply. So actually liver is little bit blessed than all other organs, because liver receive a blood supply from two sources. The first is the hepatic artery, which deliver oxygenated blood from the general circulation, and the second is the hepatic portal vein, delivering deoxygenated blood from the small intestine containing nutrients. Why I have added this point, we will discuss it. We will discuss more into it, why it is blessed. Because since there are two type of sources, so when we go for the targeted therapy, so if we block one of the arteries, then what are the conditions or what are the things, difficulties, drawbacks that we get? Since there are two sources, we can do that, which is a blessing for us. So liver, since there are two sources, we can do this therapy with the liver. Then there are four major liver cells: hepatocytes, hepatic stellate cells, Kupffer cells, and liver sinusoidal endothelial cells. Now, coming to the functions of the liver. So, so I don't go much details into the hepatocytes, because this you have already studied in your school and in your degree also. So what are the functions of the hepatocytes, what are the functions of the liver cells, but just I would like to read out what are the basic functions of the hepatocytes. The basic functions, they are involved in the synthesis of the cholesterol, bile salt, then fibrinogens, all the nutrients. And endothelial cells, they are the scavengers. Then Kupffer cells, which is very, very, very important, because the Kupffer cells, they store liver lysosomes and they digest and dispose of dying cells, unnecessary proteins and foreign microbes. So they are main immunogenic cells of the liver. Then we come to the hepatic stellate cells. So hepatic stellate cells are the dominant cells, liver cell population, which store vitamin A, which is very, very important, because I'll be telling a little bit about vitamin A in the later part of my discussions. So vitamin A, and when stimulated, promote stress response and antibody productions. And there are, of course, other cells. Now we come to the stages of the liver damage. So as we see, so these are the stages of the liver damage. We can see, laser or something?
Laser,
okay.
Laser will not work on this.
Oh, yeah. All right. So, okay. So as we see, this is the healthy liver. So the first stage of the damage is the steatosis. So after steatosis means whenever the liver that means healthy to fatty. They are moving from healthy to fatty. The first stage is the steatosis. Then we come to the steatohepatitis. After that, fibrosis. Fibrosis means the scars are there in the liver, and which may lead to the cirrhosis that we call as the end stage cirrhosis, and then cirrhosis lead to the HCC, that is hepatocellular carcinoma. Not all cirrhosis lead to the HCC. Few may lead to, and few may in few cases it may stop to the cirrhosis, because the patient sometimes collapse, because that cirrhosis is the last stage of the liver damage we tell. So from cirrhosis there are few which move to the hepatocellular carcinoma, the person suffering from the liver cancer. Okay, now what causes the liver cancer? There are various ways, like fatty liver disease or some genetic disease. That can be the cause. Then second is type 2 diabetes, which is very, very important. Type 2 diabetes may lead to the liver disease, liver cancer or liver disease. Then if a person is suffering from, suffers from hepatitis B or C for consecutive times, and if care is not taken, then there are high risk that it may lead to the steatosis, or it may lead to the cirrhosis or HCC. And alcohol consumption, so I should not say about this because this is very known fact, and the project that I have worked staying in Royal with my students of PG 2 years, from there we got the statistical data, and we have seen that here in this case the statistical data reveal us that the most affected age group, and it is very, very unfortunate that the most affected group is between 15 to 30 for liver HCC or liver disease due to alcohol consumptions. Then of course obesity, smoking tobacco, exposure to certain chemicals, then over-the-counter drugs, I would like to say something about it, and unhealthy lifestyles. So over-the-counter, what are over-the-counter drugs that we can go and purchase without any prescriptions? For example, in India it is very, very, like available pantoprazole, then thyroid medicines, and then there are few others. So, and surprisingly, when I went there, I saw that here even we can buy antibiotic. So like amoxicillin, whenever I was having some sort of congestion, I used to purchase amoxicillin and take, which is very, very dangerous. Because in US we don't get any of the antibiotic over the counter. And even if you are taking antacid or like acid refluxer, we have to like go for many through our prescriptions. We are not. Because these over-the-counter drugs, they affect the liver to a greater extent. And of course, the last point is the unhealthy lifestyle. We all know that we are living a very unhealthy lifestyle, like we are not working, our work is more with the brain than with the physical things, and then we are late nights. And yeah, I would like to add one thing with due permission of our sir. Sir, I have seen that the Western people, they are taking our good cultures and we are bringing the bad cultures. So here when we come, we see lots of KFCs, McDonald's, but their people are taking our food. They are using millets, they are using, all those good grains, which were basically the food of the Indian. And you will be surprised to see how the people are doing yoga there now. So even the Indians who never did yoga here, we are going there and doing yoga. So that is the difference. Like a healthy lifestyle is very, very important. So unhealthy lifestyle, liver is affected most. Okay. Now, what I discussed was about the different stages of liver disease. This I am concentrating in the stages of the hepatocellular carcinoma. That is the liver cancer. So, say if this is the healthy liver, which has the reddish brown color and little fat stored, and then we come to the the fat start accumulating in the liver. After that, the fat accumulation, they cause inflation and their injury start, that is the mass. And then it comes to the hepatocellular carcinoma. So when you can see, in my next slide, you can see that how this liver, this is the structure of the liver cancer, hepatocellular carcinoma. So if a person is suffering, the condition becomes if you see you can easily see what is the condition of the liver at that time, the person suffering from the hepatocellular carcinoma. Now there are certain stages of the hepatocellular carcinoma. So if you see the first stage, there are 4 stages. So the first stage, when it initially starts, and it is very, very unfortunate that we don't get the symptoms in the first stage. We never get the symptoms in the first stage. So when we get the symptoms, it is already after the metastasis, we start getting the symptoms. So we start getting the symptoms only when it starts metastasis, that is this position. From 3B, when the tumor invades into the larger vein liver, we start getting the symptom from that time. Before that, there is no symptoms. Okay, so it's like a silent killer. You don't know what is going inside your body. Alright. Now, how they are diagnosed? So there are few clinical ways. First of all, the blood works. So there are certain blood tests which include complete blood count, then platelets count, liver function test. So from liver function test, when we see that they are not working, that there is some elevated level of SGOT, SGPT, we go for certain other tests. And then we go for abdominal ultrasound, CT scan, MRI, and ultimately the liver biopsy. Usually I have seen one thing, the line of treatment is very different because personally I would like to say I know more about liver cancer because my mother died of liver cancer. So the treatment that she was going through here and the treatment that I saw there, it's very different. And there immediately they don't go for biopsy. They go for certain stages first. one after another. Because they tell if there is biopsy after the liver, immediately it starts, like sometimes it happens that it starts spreading. So they don't do that. But yes, of course the biopsy is the confirmatory test. If all the tests are showing if that the markers are high and all other parameters are indicating towards HCC, yeah, we have to go for the biopsy. Okay. So since I have already said that we don't, in United States there is one thing that like annually we go for routine checkup, which is not possible in India because of its population. So there everybody, like those who are insured, and more or less all are insured there, so everybody has to go through a screening, annual screening. In that case, if any abnormalities comes, then they go for the test, but which is not possible in country like India. So, and of course there are no symptoms. So when when I say, as I have shown that when the symptom starts, it's 3A stage. So 3A or 3B. So then what are the symptoms? The unwanted weight loss. Like if you are losing weight by your choice, by dieting, by gym, that is different. But if you are losing weight, like unwanted weight loss, suddenly the person loses the muscles and become very thin. Then blood in the stools, and then blood vomiting is there. Of course, as I already said, the jaundice, hepatitis B and hepatitis C. Then fatigueness is there, yes, ascites. So ascites is the last stage of the liver of carcinoma, because whenever the liver is after steatohepatitis, then they go to cirrhosis. So when cirrhosis is there, there is accumulation of the fluid in the body. So that we call as the ascites. And then, this is the last symptom, which is very, very dangerous, that is encephalopathy. That means in the advanced stage of the HCC, the person feels like hallucinated, or like in a dreamland or something like that, cannot say. That we call as the encephalopathy. So that is the, if such things are happening, then it is the very, very advanced stage of the HCC. Yeah. Now, what are the treatments? I would like to discuss more about the treatments. So the treatments, of course, surgery is there. So by surgery, you can remove the tumor, and there are surgeons who perform both traditional and minimally invasive laparoscopy are there. Then transplant is there. Then ablation techniques are there. Then this involves either surgery or it may be done by the process of the radiology. And intra-arterial therapies, which I'll be showing you in details, and of course chemotherapy. Now this is the stage, this is intra-arterial therapy which we are doing. I have been shadowing the doctors while they are doing the experiment. So this is what we do, this is our lab. So there is a change in the lab from biological to the clinical. So this is what we do in the lab. In the clinical research, how they do the techniques, I would like to tell you. Okay, so there are certain techniques, we call them as first is mapping. Now what is mapping? So there is the invention, we put the catheter and we take a structure of the arteries and where the tumor is actually located, and the structure of the arteries are there. So that is what we call as the mapping. We get a clear picture of the liver of the patient, that which portions are affected or where is exactly the position of the liver, how we have to proceed. Then it is TRACE, which we call as the TACE, we call as transarterial chemoembolization. So what is chemoembolization? Instead of giving a general chemotherapy, what we do, we take the medicine, we are using doxorubicin here. So now doxorubicin is I will tell you in the next slide that what is doxorubicin and how it works. So we take the medicine and we do a little incision and then put a catheter inside the patient, and then we reach the tumor spot. And after reaching the tumor spot, that doxorubicin is released exactly in the tumor. that process we call as the TACE, that is chemoembolization, so that it can work on the tumor itself and it can stop the metastasis process. Or sometimes we can cut off the blood supply. That's what we were telling, because since the liver has two sources of the blood supply, we can do that experiment with the liver. So cutting the supply of the blood to the tumor. As a result, after some time, the tumor goes for necrosis and dies off instead of going for the metastasis. Then there is a TARE. TARE is transarterial radioembolization. So here we use instead of the doxorubicin, we use the radioactive substance which we put there, and then we restrict the growth. It's almost, TARE and TACE is almost, the process is almost same, but the content or the chemical what we use is different. There we use doxorubicin or other chemical, and here we use the radioactive substance. Then it is SIRT, where Y90. This Y90 is a very, very popular treatment which we are giving to the patients. So Y90 is a radioactive element. Now Y90 is after the same process, we put the Y90 there. But then you may ask me that if it is radioactive, it may affect the entire body. No, it has a region up to what it will show its radiations, and that is very small. That is very small, and it will restrict to the tumor, not to the other part. Then, there is a blend embolization, which is very, very important again. So in blend embolization, it is used to treat the liver for post-surgical treatment. After, the patient is having transplant, or after, there is a removal of the tumor, we go for the blend embolization. Now, these are the few medicines that we are working with in our lab, in the basic lab, that is sorafenib, and then doxorubicin, nalfenavir, and isrin. Now doxorubicin, it is very, very commonly used medicine in HCC that is widely being used. Now, what is doxorubicin? Before doxorubicin, we will go to the sorafenib. The sorafenib is thought to exhibit a dual mechanism of action, and it blocks the tumor proliferation and growth by inhibiting the RAF and extracellular cell signaling, signal-regulated kinase. So this is how sorafenib acts. But this is an established drug. This is already used. Then we come to the doxorubicin, which is very, very important, and we are widely using doxorubicin. Presently we are not using sorafenib because that is used in case of the initial stage, or the person who don't go to the advanced stage, we give them and there. But when the patient goes to the advanced stage, we use the doxorubicin. Now doxorubicin is a very, very important medicine, and there are two proposed mechanisms by which doxorubicin act in the cancer cells. Because number one, it intercalates into the DNA, and disruption of the topoisomerase mediated DNA repair. There is a twist, and the generation of free radicals and their danger in the cellular mechanism. Now what happens, since it acts by intercalating the DNA, but there is a DNA repair pathway. So it has been seen the person who suffers from HCC, in that person there are two factors which are increased. That is HSP70, heat shock protein 70, and APE1, that is anti-base. So these are, and now when they are elevated, they combine together and they help in the base accession repair. As a result, they put a negative feedback to the doxorubicin. When they are putting a negative response to the doxorubicin, then instead of going to the apoptosis, it goes to the cell growth. So it doesn't work. Sorry. So it doesn't function properly because of this HSP70 and APE1. So what we are doing in our lab, so we are using a protein X pump, which the function of that protein X pump is to stop the combination of the HSP and APE1, because it is seen that they increase by 100 folds. Now when the combination, or when the HSP and APE1 is separated, they cannot work and they cannot help in the base excision. As a result, the doxorubicin can function. So this is one way how we can make the doxorubicin functions. Secondly is the nalfinavir and istri. These are the two drugs that we are combining. So they work on the ER stress. Nalfinavir and istri both work through the ER stress. So we have thought for a secondary mechanism or secondary pathway so that we can avoid the doxorubicin, and those are nalfinavir and istri. Now nalfinavir and istri, they work through the ER stress. Now it is seen that nalfinavir and istri, they may not function properly individually, but when they are combined, the combination of nalfinavir and istri is fantastic. So they help in the cell apoptosis. So this is our, this is what we have done. We have done the MTS assay with N1S1 cells and also the Mori cells. There are two types of cell line cultures. One is suspension, another is adherent. So we have tried with both types of cell culture, cell line, and it is said that nalfinavir and istri, their combination is effective. So you can see how it has led to the cell apoptosis. This part. Alright. So here in Cleveland Clinic, like there is a liver cancer program, which is like designed so that all of the special, what are the like line of treatment or how we do. There, like when a patient comes, it's like a like there is a liver tumor board. So the patient's cases are presented there, and there are different type of physicians who work on that particular patient's parameters, and then they decide that what we have to go. It's not that one patient is coming to the doctor and doctor is doing whatever he likes or what is. So it's after the discussion. So there is a that is a very good program in Cleveland Clinic Cancer Program, that it is designed so that all of the specialists, they work as a team. And this allows you to save like time by eliminating multiple appointments. You don't have to go through. And then, of course, after coordinated care in one sitting, the expert then develop the individual treatment plan. And here there is no general treatment plan. That is the best part I have seen there. Every individual has their own physiology and their own like genetic makeup, and so every individual, so it is an individualized treatment for every patient there. And this one is awareness like slide for all of you, because see, liver is very, very important. It is the top like detoxifying organs. So this may lead us, these are the things which lead us to the liver disease, and these are the things of unhealthy lifestyles. So if you forget other slides, I don't mind, don't forget this slide. Alright, so this is my, like the lab where I work. This is Lerner Research Institute. Oh, keep that with this slide. Ask any.
If you have any questions, then you can.
It should not be if, it should be you should. You ask questions. Yeah.
Another question is that when in the site of the tumor formation, when do the blood vessels get expanded or show any structural changes? Because will they show?
Yeah, because if there is tumor formation, then that portion, I have already shown one slide. So you see, if you go to the first slide, this one, you see, it's already deformed. You can easily, and you'll be surprised, I'm sharing my personal experience, like when we were experimenting with the human tissue. So it was surprising, like when we got two types of, one is non-treated, another is treated. So the treated one, the person suffering from HCC, it's so much like soft and damaged, like I was scared of holding that this is the human tissue. So it's very surprising that like the normal liver tissue, it's very well, you can cut it and you do everything. But when it is like, when it is treated, it's like it's almost ripe fruits. It's very unfortunate. Yeah, there will be changes, and of course, like because the tumor will go for metastasis and then they start reducing. And yeah, there are another thing I would like to add, like sometime, say this type of like chemo tear, what I have discussed, these all will be, more effective when the cancer is in the liver. When the cancer is in the liver. But sometimes it happens that there is metastasis, then it moves to the liver. For example, from breast to the liver. So in that case also, these treatments are applicable, but there is no guarantee for the metastasis part. On the other part, it is already spread. But the liver part will be, restricted in that case. But chances are not that much, it's not 100% chance of success. Because it is already metastasized. There is already metastasis, and it is spreading to the other parts. We are controlling the liver, but other parts we cannot control. But if it is in the liver, it is 100% successful.
And that means when the catheter is used for the mapping stage, and that means only after a certain stage it will be actually applicable, right?
Which one?
The mapping part.
You are talking about mapping?
Yes, ma'am. When we try to detect the region of the tumor happening, so that means only after a certain stage of proliferation only it can be used.
See, when I have shown you the stages. So the stages. Now you see, as I told that there are the symptoms. The symptoms will start coming from the stage B, 3B. So when the symptoms are there, immediately the mapping is done. So when the mapping is done, it is shown that which part of the liver and what are the arteries supplying blood to that part. So mapping is just getting a structure of the tumor and its surrounding vessels from where it is getting the supply, so that we can work on that particular vessel and either we stop or there is a disconnection to stop the blood flow, or we give the TACE or or we go for the chemo or the radioembolization to restrict it, so that it goes for the necrosis and there is no further metastasis. Yep. Yes.
Is it directly like the tumor induced?
No, it is directly, the Y90 we take in a low dose and we put in the beads or the catheters. There are two ways. One is DAB, we call it DAB, drug-eluting beads. So there are drug-eluting beads, we put it in the beads. So sometimes it is seen that beads, instead of putting it without the beads, is more effective than if we put it through the beads. Because in beads what happens, when the beads are released in the tumor, they release it in a very slow way and for a longer time. So it's so that it becomes more effective. And as you are telling that it is taken through the catheter, not like
After the treatment is done, it is
no. It stays there, it stays there because it's radioactive. Now after some time it will goes off and then you don't have to take it out.
Ma'am, how can we sense our field from zoological field to clinical field?
Yeah, that's a good question. That's a very good question. See, zoology, as I already tell that it's a subject where you have medical science, you have ecology, you have biochemistry, everything. It's a mixture of everything. So basically we know or we learn the things. Now the thing is that if you are I give you my case. I am not something from medical science or something, but I am basically a fish physiology and biochemistry specialization. So I have been doing work. Now when I was trying, there is nothing, in US, there is nothing that you cannot do anything. If you are interested, you can move there, you can apply there. Suppose you are doing PhD on certain things. So on what thing you have done PhD, so if you try on applying that, you'll get. But if you are interested in something else, you can also get there.
Without doing PhD, how can I?
No, without doing, no. There is an integrated course. If you want to do PhD there, that also you can do. But after PhD also, you can go for postdoc. So you can apply. It's not that. They will take your, in my case, it was working in the liver. So how my shifting occurred, after fish physiology and all those, I started my project in the liver cirrhosis. So that was a demographic study which I did with the PG students of Royal Global University, first and second batch. So we collected a huge number of data. Our sample size was more than 1,000. So in that case, we got a good number, and with this only I started applying. So it was like a plan for me that I will do something on that so that there is a connecting link, and you go that there. But it's nothing that if you're from zoology or botany, even one of my colleague who is a master's in botany and she is from JNU, she is in this line. So it's nothing like that. We can apply. If you want, my email ID is there and contact number is there. Anyone, if you want, you can contact me, and if I get any fellowship or anything that like exchange, student exchange fellowship or something like that, I'll definitely tell it to Dharmesh so that he can circulate it among the students. You can also apply for that. If you want to do PhD there, it's all right. If you want to do PhD here after PhD also, you can apply. There was someone. Not only HCC, any type of liver disease.
Yes, sure. Now, apart from those, is there any other factor which can still lead?
Yeah, of course. See, that is not in my mother died of liver cancer. Yeah, and she had breast cancer, after that liver cancer. So in there, when I went there and when I was talking about this, then they immediately went for all those tests. So they did the test in me. See, there I don't have anything to do. That is genetic, completely. But we don't have any, that is unfortunate. We don't have anything to do with that. Even though we live a healthy lifestyle. But yes, healthy lifestyle is in our hand, we can do that. Okay, there are other factors. Genetic factors are there. Then diabetes are there. Of course, diabetes also I consider as a very unhealthy a disease of maybe of genetic and unhealthy lifestyle. So these are the basic two factors. So genetic factors you don't have, that's not in our hand. We cannot do anything. But of course, yes, healthy lifestyle we can.
Good morning. Ma'am, after stopping the blood to flow into the tumor, you told that necrosis will happen. So it will completely cure the tumor, or if there is any chances to further?
No, of course there always remains a chance of they will, because that tumor may not, but there are some surrounding cells, they may again aggravate and they may again form a tumor. But that tumor surely go for necrosis. From there is no chance of metastasis. Anyone else?
You said since the symptoms only appear in the stage 3 right? So is there some way of detecting it earlier and preventing it?
No, that is the main thing, because here we don't go for annual screening. What it is done in United States. But yeah, of course, we can go for regular checkups so that we can stop it. And of course, we can go for regular checkups and.
So it is possible to check up?
Yeah, it is possible, because there are the test labels, and until and unless you go to the extreme stage, there is no, there will not be biopsies. But of course, blood parameters, liver functions, if your liver functions are same, then there is nothing wrong in your liver, so then it's fine.
And then one more question. Why do you consider too late for treatment at what stage?
You see, in US there is no stage which is considered as too late. Because every stage is late, and in every stage we can work. Okay, but yes, of course, the last stage we call it is as this one, because it has already, when it spread to the distant organ, that is metastasis, then you cannot do anything, because it has, from here it has moved to the distant organ, that is the brain. This is the last stage, where usually there is no use of, we cannot help. And that stage we call as the encephalopathy. So the person already started forgetting because they have already metastasis.
So that's why when the brain impairment.
Yes.
The
Hello ma'am. Ma'am, my question is that which age group has more chances of getting infected by these cases?
Huh?
Which age group has more chances of getting infected by these cases?
See, if it is alcohol, my study that I have done, it is from the age group of 15 to 30. But there is no like nothing, because we have seen children also suffering a lot from HCC. It may be genetic factor, it may be some other factors, and also that 85 years, 90 years, they are also suffering from. So there is nothing age group, but yeah, of course, if it is unhealthy, it is definitely from 15 to 40 years.
Thank you.
Who else?
Madam.
Yes, sir. I'm waiting for that.
I just wanted to know the methodology for treating is catheter-induced radiology. There are liposome-induced radiology tools also.
Yes, sir.
So which one is more effective? Is it the liposome-induced? The chances over there is that the tissues can be disrupted, and that can lead to what we call as metastatic phase infection. If it is liposome-mediated drug delivery, there is no catheter involved. Which one is better?
Sir, that is the traditional way we have. That was the first type we have. It was started. That is the first line of treatment given. But sir, now there are some drawbacks for that, and like in the previous one, the chances of metastasis is more. So we considered this one to be more effective. Though there are drawbacks, but still effective is this one. Okay.
Is it that the delivery aspects are more
Sir?
effective here, or
Sir?
is it the delivery of the drug is more effective here, or is it
It is more effective here, sir.
the delivery is more? Second question is here, are there any differences in the protein profiles of the membrane,
Yes.
cancerous cell and non-cancerous cells?
Yes, sir.
There are.
There.
So can we not have targeted delivery to liposomes, targeting those differences in the membrane?
Yes, we can, sir. But is she fine?
Okay, we can go.
Sir, I would like to ask again.
So what I was saying is that if there are differences in the membrane profile, can the targeted delivery liposome-mediated delivery not be very specific to
Yes,
target?
it is very specific, sir.
Very
It is very specific.
Okay. Continue. Thank you, madam.
Not one last question.
Yeah, sure.
I think recent studies have shown that there is a very strong relationship between the heart and the liver. So I think early symptoms of diseases of the liver also affects the heart. So have you done any kind of studies relating these two? Or it would suggest like if someone is having some kind of heart problems, should we also consider diagnosing the liver?
We have not done any such work like that. But yeah, whenever there is whenever you are going for any cardiac treatments or there is here also we go for all tests. So that is the reason to find it out. But we have not done any such work on that, like heart and liver.
What a fantastic deliberation. Very important points were discussed today, and I want to thank each one of you for being such patient listeners to learn about hepatocellular carcinoma and its targeted therapy. I would like to invite our Dean Sir to suggest a few suggestions for our students.
In fact, this is something which I had told Ramesh, now I want to speak. Earlier it was just a formality. What made me tell Ramesh that I want to speak is a question which had come, that as a zoologist, how can you, there was some question like that, how can you consider some medical, what was the question?
From zoology to clinical.
How can we change our field from zoological to clinical?
It's all here. You got my point? The first question that's important is that is there a target in front of you? A person who has done fish biology from Guwahati University, long back, and if you go for a job, you will think, okay, अब क्या है, अब तो job तो मिल गया, अब मछली पकड़ते हैं, देखते हैं क्या हम fish ecology, fish biology, or maybe we will do some stress related things in the fish, because that is my domain, I can't think beyond that. What's important is that if you think beyond the horizon, that is what I have been insisting to the students, that from the studies that you have, first thing, always think that the concept when you are in the class that the teacher is fooling you. So you will try to catch hold of your teacher that you are wrong, sir, you did not tell us this thing. But that will happen only if you study before the class, and by that your horizons will improve, your capacity to think will improve, and that will make you identify targets. That you are doing MSc Zoology, what after MSc Zoology? What is something great in zoology? What is innovative in zoology? Or what is different that you have not been able to study in zoology, which you would like to come? Some people may say that, oh, I like to conquer and go up our Everest, but the target. So if you think that I am a zoologist, so what do we do? What my teachers tell, I do that because there is nothing else to do, then the world is gone for you. What's important is that you set your targets. And targets are not to be set when you are in your 4th semester. Targets are to be set when you enter your university career. Not that you did not get a seat in biochemistry, you would go to zoology, or you did not get a seat in zoology, you will get a biochemistry, or any other subject, or if you got nothing, you will do A, B, C, D. The moment you are in that type of situation, you are gone. The moment you do not set your targets, you are gone. Life is full of challenges ahead of you, provided you understand the challenges and you understand the solutions to the challenges, probable solutions to the challenges, and the targets that you can fish out. There's an example, a student's project led her to something different. So we are all giving you projects, all your teachers must have given you projects. How many of those projects have led to one publication? Has anyone? The answer comes straight. If it has not led to any publication or any further study, today you do on rat, tomorrow you do on mouse, third day you do on something else, fourth day you do on brain tissue, the teacher keeps on giving different projects to different students, and also RAN work goes on. If it is that, then the question itself is a big question, because you think as a zoologist I cannot become an oncologist, or I cannot become even a chemist, and you need NEP to become that. The problem is not with the education, the problem is within us, that we are not able to understand what we want to do. I was in an orientation program for PhD, classwork, dash, blah, blah. I don't believe in all that shit. What is important is that you identify a target and try to work towards achieving that target, even in your MSc. If you are able to do that, you do justice to yourself. So the question itself says that you think as an MSc zoology guy, you cannot do anything beyond what is being taught to you. That's not the case, whether it is India or whether it is US. What's important is that you understand what you want to do. And I hope you understand what you want to do, but I know you do not understand what you want to do, because my own experiences have been here. Nobody is perhaps coming up with challenges. Even in the School of Life Sciences, People are not coming up with challenging questions, challenging solutions. The moment you start coming up with that, you will be good. And I would assume that you inculcate that type of approach in you. With regard to what Sushmita said, it was a very good talk, defined, targeted work that we are carrying out. There is much more on the backside which she must not have told you because of the paucity of time and also the nature of the audience. But I congratulate her, and on behalf of the School of Life Sciences, congratulate her for the work that she has done. Thank you very much.
Thank you, sir, for your valuable suggestions. To conclude the session, I request Dr. Anukumari ma'am, Assistant Professor and Event Coordinator in the Department of Zoology, to kindly deliver the vote of thanks.
A very good afternoon to each and every one of you present in this August session. On behalf of the Zoology Department, the Assam Royal Global University, I, Dr. Anukumari, extend heartfelt gratitude to all for gracing us with your presence and making this interactive session an outstanding success. First and foremost, our sincere appreciation goes to our esteemed Dean of the Royal School of Life Sciences, Dr. Professor Nikhil Kumar Churungu sir, for taking the time from his busy schedule to enlighten us with his presence. Your support and encouragement means a lot to us, sir. And thank you for the suggestions that you have given at the last moment. Thank you so much, and I hope that all the students must have listened to that. Like sir said, please set your target and all. Thank you, sir, for that one, for inspiring our students. I am deeply thankful to our esteemed guest and the founder of our Zoology Department, Dr. Sushmita De, Madam, for traveling all the way from the USA to share her individual insights on the topic Hepatocellular Carcinoma and its targeted therapy and overview. Your expertise has really enriched our understanding, Madam, and we are grateful for your presence among us. Ma'am has talked very well, and as sir was saying, she's ever smiling. I can see her smiling right now also. So it's good, like you take some challenges and then like ma'am is smiling. Sometimes I used to be like how people they used to manage that kind of smile, so I have to learn from her. A heartfelt thank you to each of my colleagues and seniors present here for their unwavering support, especially to our head, Dr. Dharmeshwar Bharti sir, whose guidance has been instrumental in organizing this wonderful session. Sir was doing up and down, arranging everything, so thank you, sir. A special word of gratitude goes out to our dear students whose enthusiasm and passions have infused the atmosphere with energy and contributed to the resounding success of this event. I extend my heartfelt thanks to Disha Agarwal for being our host for the day. I told her at the very last moment and still she was ready. I'm happy with you. Last but certainly not the least, I extend my sincere appreciation to the IT department for their hard work, dedication and perseverance in ensuring this smooth conduct of this session. I apologize if mistakenly I have missed on anyone. And once again, thank you to everyone who has played a part in making this session fruitful and memorable. Your presence has indeed made a difference, and we eagerly anticipate welcoming you again in our future endeavors. Thank you.